Hello, my name is Julie Bourgeois, and I am a third-year graduate student in the Interdisciplinary Biological Sciences program here at Northwestern University, where I work under the guidance of Professor Chad Merkin. I completed my undergraduate Bachelor of Science degree at SUNY Polytechnic's College of Nanoscale Science and Engineering, where I majored in nanobioscience. Nanoscience involves the study of materials and particles that have at least one dimension between 1 and 100 nanometers in length. Nanoscience positively impacts many disciplines, spanning physics, chemistry and engineering, to biology, the life sciences, and medicine. The nanoscale is often referred to as the natural length scale, and it is particularly relevant in the context of living systems. Nanoscale features are responsible for interesting biological phenomena, from the extreme hydrophobicity of forest leaves to the iridescence of a butterfly's wing. Size compatibility is very important. For example, the DNA double helix is approximately 2 nanometers in width. Proteins are, on average, 10 nanometers wide. Viruses and bacteria typically are 100 and 1,000 nanometers in size, respectively, and a human red blood cell is approximately 7,000 nanometers across. A variety of nanoparticle constructs have been developed to understand the biological world and to change how it works in the context of medicinal applications. In nanoscience, DNA can be repurposed from its biological function as the blueprint for life to serve as a designer type of glue. DNA has many advantageous properties for this purpose, including its nanoscale size of 2 nanometer width and tunable length based on sequence and number of bases, as well as highly specific base pairing. These predictable Watson-Crick base pairing interactions have been exploited to program nanoparticle assemblies into larger structures. For more information on how DNA can be used to make modern materials, please watch the previous 3 NUChem videos in this collection. These videos talk about modern material research connections pertaining to programmable atom equivalents, or PAEs, bonding with PAEs, and PAE design rules and phase diagrams. Here, I will discuss how DNA is not just a smart glue, but how it can be used to make nanomaterials that interact directly with living systems to change how they function or to detect or treat disease. In the Merkin Group, we invented and are developing oligonucleotide modified nanoparticles known as spherical nucleic acids, or SNAs. SNAs consist of densely packed and radially oriented oligonucleotides arranged around a nanoparticle core. When comparing three-dimensional SNAs to the analogous one-dimensional linear nucleic acid structure of the same sequence, many properties emerge that make SNAs beneficial for use in the life sciences and medicine. For instance, SNAs bind complementary strands of DNA both on and off nanoparticles with higher affinities than nanoparticle-free linear strands. These transition-involving SNAs are therefore sharper and occur at higher melting temperatures. These properties are indicators of cooperative binding and greater duplex stability, respectively, when SNAs are involved. This is interesting from a biochemistry standpoint, but these properties also translate to the use of SNAs as high-performance probes in extracellular biodetection schemes that have increased target sensitivity and mismatched selectivity compared to the state of the art. Furthermore, unlike their linear oligonucleotide counterparts, SNAs are rapidly internalized by cells in high quantities, over 60 different cell types to date, and they do not require auxiliary and often toxic transfection reagents to cross cellular membranes. This occurs through a process called caveolin-mediated endocytosis, where the cells engulf the particles in intracellular vesicles called endosomes. Small amounts exit the endosome, which can be useful for detecting or regulating events in the cytoplasm. Endocytosis occurs because SNAs bind scavenger receptors, which are proteins on the cellular surface that regulate uptake, more effectively than linear nucleic acids of the same sequence. Furthermore, the dense arrangement of oligonucleotides protects the SNAs from nuclease degradation. This leads to prolonged circulation times and the site-specific accumulation of SNAs as compared to linear oligonucleotides. When these properties are coupled with their higher binding affinities, SNAs, in the many forms that they can take, are uniquely suited as agents for intracellular gene regulation and immunotherapy. In the case of gene regulation, SNAs can be used to deliver both RNA and DNA to cellular compartments, allowing for disease treatment on the genetic level. Using what is called an antisense pathway, SNAs prepared with DNA strands complementary to a messenger RNA that codes for a cancer-causing protein, enter cells, bind that messenger RNA, and like a rheostat, downregulate its ability to make the troublesome protein. This method has been shown to be particularly effective for a variety of oncoproteins, including ones associated with glioblastoma, a particularly aggressive form of brain cancer. In this example, SNAs penetrate the blood-brain barrier and accumulate in the tumor environment to sufficiently knock down protein expression and increase glioblastoma sensitivity toward therapies that induce cancer cell death, where the same nucleic acids in their linear form do not. These constructs have been used in patients and are actively being developed as therapeutics for this terrible disease, as well as others, including skin cancer. With regards to immunotherapy, there has been a large push to generate immunostimulatory cancer vaccines to allow the body to recognize and attack cancer cells in the same way that it does other foreign invaders, like certain viruses or bacteria. Currently, conventional vaccines have been prepared by simply mixing the adjuvant, which can be an immunostimulatory linear DNA sequence, with a target disease-specific antigen, and injecting it into muscle tissue. This blender approach to vaccinology fails to take structural parameters into consideration, and it is ineffective in many cases. In the case of the modern mRNA vaccines, like those used for COVID-19, the mRNA strands required to make the antigen are delivered to your cells via a lipid nanoparticle. SNA-based vaccines take a different approach. They take advantage of the nanoscale structure of the vaccine components using principles of rational vaccinology. By radially arranging immunostimulatory DNA and target antigens together on the surface of a nanoparticle at high density, SNA vaccines cause an enhanced immune response against both cancers and infectious diseases like COVID-19. The structure of the SNA allows it to interact more effectively with toll-like receptors on cells that control immunity than linear nucleic acids delivered with the blender approach. Not only that, the specific arrangement of the adjuvant and antigen on an SNA can be varied, and researchers have found that they can change the potency of vaccines from being ineffective to curative. Rational vaccinology has been used to make SNA vaccines for 9 different cancers to date, including triple-negative breast cancer, one of its most aggressive forms. Furthermore, the SNA platform is now the basis for over 1,800 commercial products, including 7 drugs in human clinical trials for both gene regulation and immunotherapy. This highlights the applicability of the platform to treat diseases and solve issues in the real world, as well as touches on the overall modularity and versatility of the platform. Since the core of the SNA and the shell are entirely independent from one another, new SNA probes and drugs can be formulated by simply changing the strands on the surface or the material of the core, allowing for rapid drug development. In my own research, I use the SNA platform to treat common but debilitating neurodegenerative diseases like Parkinson's disease. Parkinson's disease is marked by the aggregation of a protein known as alpha-synuclein in the brain. Furthermore, it's been shown that the mutation in another protein, GCase, accelerates the aggregation of alpha-synuclein, thus contributing to the severity of the disease. My work is focused on using SNAs with a GCase protein core as a replacement therapy in the brain to reduce the levels of toxic alpha-synuclein aggregates. What I love about research is that it is for the curious. I love to learn about how things work and why certain processes happen the way that they do, especially in the context of biology and medicine. Every day I strive to learn something new so that I might be able to move not just my own research, but society forward. I was fortunate to start my research early on in my undergraduate career, and it has prepared me very well, not just for graduate school, but for life and a future career in the biomedical field. If research is something that you are considering, I highly recommend beginning now as an undergraduate, as the skills and techniques that you learn will help you excel in all of your future endeavors.